Good morning, everyone. Couple quick announcements. So we have an exam on Monday, as you know. If you check on BSpace, I saw that Mike had posted the room assignments for that, so check it out. I'm holding an exam review session on Friday night, 7 to 9 p.m., this Friday night in Dwinell Hall. It's strictly a question and answer, so I'm not going to be lecturing at all, just answering questions. So if you want to come to that, it's going to be in that big lecture hall in Dwinell Hall, 7 to 9 p.m. Friday night. Also, I'm planning to post one or two more problem sets. These are ungraded, so check BSpace for those on the topics that we've been covering last week and this week in this class. And again, those are just for your sort of study guides as you're going through the material and getting ready for the test. So this week, we're going to talk about photosynthesis. And this is chapter 10 of the book. And what I want to do, so we have two lectures on photosynthesis, and what I want to do today is primarily to talk about light energy and how cells harvest this and convert it into chemical energy. And so we're going to focus on the light reactions. And then on Friday, we'll talk about how cells actually use that to synthesize molecules. Can everybody hear me? I feel like it's quiet. Can you hear me in the back? Yeah, good? Okay. All right, so basically, you probably know that photosynthesis is the process that converts solar energy into chemical energy. And basically, this is how essentially all life is possible due to this, right? That we're basically dependent on harvesting energy from the sun and then ultimately converting it into organic molecules that are used to drive all of the cellular processes that go on. And so what we want to do today is talk a bit about how that actually works and how light is harvested, and some of the energy that comes in is actually captured in the form of chemical energy that can then be used later for synthesizing more complex molecules. And so we can start off by just some definitions. So if we talk about autotrophs, these are organisms that sustain themselves without having to consume organic molecules for food, right? They're basically able to synthesize all of the molecules that they need to live. And so if we're talking about plants or other kinds of organisms that are capable of photosynthesis, these are autotrophs. They can harvest energy and use that to synthesize the molecules that they need. And these are basically the producers in the biosphere. They make organic molecules from carbon dioxide and other inorganic molecules sometimes, and can incorporate those into more complex molecules. So most plants are photoautotrophs. They use the energy of sunlight to make organic molecules from water and carbon dioxide. And there's some interesting examples of photosynthetic organisms that use something other than water in that pathway as well. We'll just touch briefly on that. And the book has some pictures of different kinds of photosynthetic organisms. So plants, of course, but also some unicellular protists are capable of photosynthesis. And we can tell because they're green and they have chloroplasts. We also have examples of algae. So this is an example of multicellular algae that have photosynthetic cells, chloroplasts. Cyanobacteria, shown here. These are actually the organisms that are thought to have been possibly the precursors to modern chloroplasts, right? Cyanobacteria that might have invaded cells eons ago and eventually given rise to the modern organelle called the chloroplast. And then finally, purple sulfur bacteria. So these are actually organisms that are using, rather than using water, they're using hydrogen sulfide for a photosynthetic pathway in which they're actually producing these sulfur nodules that you can see here, these yellow nodules. Very cool, interesting pathway that's parallel to the one that we'll talk about today. So heterotrophs, these are organisms, of course, that obtain their organic material from other organisms, like us, right? They're the consumers of the biosphere. They have to consume organic molecules. They can't make all of their own materials. They have to acquire them from their environment. And these are organisms that depend on photoautotrophs for food and oxygen. So it's part of the ecosystem. And so fundamentally, photosynthesis is the process that converts light energy to the chemical energy of food. Chloroplasts are structurally similar to and likely evolved from photosynthetic bacteria. And what we can see is that the organization of these cells allows for the chemical reactions of photosynthesis. So again, it's important to kind of think about where these processes are going on. Where are these enzymes located in these cells? Because it's incredibly important that they're organized the way that they are, right? Where membranes are set up, where we have a large surface area for these pathways to occur, which enzymes are located inside of a membrane or associated with a membrane, and which sets of enzymes are located in the soluble part of the cell, whether it's the cytosol or whether it's the part of the interior part of the chloroplast that we'll talk about today called the stroma. Right, so these are the sites of photosynthesis in plants, chloroplasts. So primarily, these are located in, and these are going on, these reactions are occurring in leaves of the plant. Their color is due to a pigment called chlorophyll. And this is the pigment that is responsible for interacting with light energy that enters the biosphere and absorbing a lot of it, not all of it, and then using that to generate and store chemical energy. Not generate it, but store it. And so the light energy absorbed by chlorophyll drives the synthesis of organic molecules in the chloroplast. So what happens is that carbon dioxide enters and oxygen exits the leaf through very tiny pores that are called stomata. So we can think about carbon dioxide coming in through these pores, and the waste product, I guess you could say, of the pathways is oxygen, and that's produced, and it exits through these same pores. How does water get in to plants? Does anybody know? What's the primary way that it gets in? Through the roots, right? Yeah. So it gets in through the roots, and it travels up the veins of the plant. So it gets in through the roots. But carbon dioxide is a gas, so that gets in through these very tiny pores, and oxygen, same thing. The gas, it exits through these pores. Here's a diagram that sort of shows a close-up of what's going on inside of these cells in the leaf. So the chloroplasts are found primarily in the cells that are inside the leaf tissue, cells of what's called the mesophyll. And typically, a cell in the mesophyll has roughly 30 to 40 chloroplasts in it. If we look at a close-up of that, what we see is that the chloroplast, the chlorophyll, this pigment, is located in the membranes of these thylakoids of the chloroplast. So these are connected sacs that are inside the chloroplast. And this cartoon shows the organization of these. You can see that they're sort of analogous to what we saw when we looked at the structure of the mitochondrion, where we had that very highly enfolded membrane that created the cristae of the mitochondria. So see a similar kind of thing here, where we see these very highly enfolded membranes. These are the thylakoids, and these are stacked up so that they form these granum, right? These almost looks like a stack of pennies or something from the outside. And slicing through it, you can see that it has an interior called the thylakoid space. That's inside these very enfolded membranes. Outside is called the stroma. And then, of course, the chloroplast is enclosed by both an inner membrane and an outer membrane. So just like the mitochondrion that has those two, the double membrane organelles, same thing is true for the chloroplast. And if we look at a micrograph of this, slicing through it, you can see, again, these very highly stacked thylakoid membranes you can see here in these enfolded structures. These are the granum right here. And so these, the chloroplast also contains stroma. This is this sort of dense or thick fluid out here. It's actually, when they draw this cartoon, it looks like it's empty, but in fact, it's packed full of enzymes that are busily involved in the photosynthetic process. So, back when people have been very interested, of course, for a long, long time in understanding how does photosynthesis actually work, how do cells in plants do this, how do they harvest light energy. And this is the overall equation that we can write for photosynthesis, in which we have 6 molecules of carbon dioxide plus 12 molecules of water in the presence of light energy combining to form a molecule of, what is that? Glucose, right? So that would be a sugar molecule plus 6 oxygen molecules plus 6 water molecules. And a couple things are worth saying here. So, they're showing you this overall reaction, but bear in mind that, that the actual product of the photosynthetic pathway is not a glucose molecule, right? So that's actually a 3-carbon sugar initially, but ultimately it can be used to make glucose that can then enter the same metabolic pathways that we talked about last week, right? Breaking it down and using it to build other kinds of molecules. So this is just to give you a sort of overall sense of the chemistry that's going on. But remember that, there's lots of variations to this. Yeah. Is the glucose actually made in the chloroplast? Yeah, and we'll look at that. And then it's transported out for other reactions, right? So there's lots of transport that's going on here. Yeah. Well, right. So this is just to try to write a balanced equation here, right? So we could just cross that out and have 6 waters shown over here. But I think the point is to show you that ultimately this is how electrons are being transferred in this reaction, right? And they want to show you that that 12 molecules are actually of water are being consumed, but 6 are actually produced in the end. Okay. Now this right here just gives an indication of what's happening to these different atoms that are in the reactants of the overall photosynthetic reaction, where they end up in these products here. Anybody know how do we figure that out? If you read the book, you'll know. But how can you do an experiment to figure that out? Anybody know? Right, isotopic labeling, right? So, and the book talks a little bit about this. So, and this is actually what was done. So the idea was to, initially, in the early days, it was actually thought that what happens in photosynthesis is that these reactions result in the splitting of carbon dioxide, right? The splitting of carbon dioxide. And it was actually through isotopic labeling that it was figured out that it's actually the splitting of water that leads to the production of oxygen. It's not the splitting of carbon dioxide that leads to the production of oxygen. Because if you use, for example, O18 labeled carbon dioxide and use that in basically provide that to a plant or to a system that's carrying out photosynthesis, then you can observe what happens to that O18, and it actually doesn't end up over here. It doesn't end up in the oxygen. It ends up in water and in the sugar molecule that's produced. And you can do this with other, you can label with other kinds of isotopes as well and follow what happens in these reactions and figure out what the pathway is. So this actually took a lot of work over many years in many laboratories to really try to figure out what the overall reaction is and how, what's happening to the different reactant components at the end of the photosynthetic pathway. Yeah. So he's wondering about the color of chlorophyll, right? And we're going to talk a lot more about that today, okay? So maybe hold that question, but he's wondering, can it be anything other than green? And the answer is that, yes, there are other pigments that do play a role in photosynthesis. They don't tend to be the major, obviously the major component. We don't see a lot of orange plants, right? But it turns out that chlorophyll is a very efficient pigment for harvesting light energy, but it does have some accessory pigments that help to broaden the spectrum that can be used for visible light. Okay? All right, so overall, photosynthesis is a redox process. We've talked a lot about redox reactions in this class, and this is another pathway that involves oxidation and reduction reactions. And overall, what's happening is that water is being oxidized, carbon dioxide is being reduced, and so we have this sort of a pathway here where we're actually, in this case, synthesizing a more complex, more reduced molecule from these reactants over here. So overall, this is an endergonic reaction. It's an energy-requiring process to synthesize these more complex molecules, and of course, the energy in this case is coming from light. So we can really break down the photosynthetic pathway into 2 phases. We first have the light reactions. This is the photo part of photosynthesis, and then we have the Calvin cycle, which is where synthesis of molecules is actually going on. So these light reactions that are going on in the thylakoid membranes of the chloroplast result in the splitting of water, the release of oxygen, and the reduction of a coenzyme. In this case, it's NADP, NADP+, so very similar to NAD except with an extra phosphate group on it. This gets reduced to NADPH in the process. And just like we saw when we looked at respiration and the pathways there, NADPH is a way of storing chemical energy for use later in the Calvin cycle. And what also happens in light reactions is that we generate ATP from ADP in a process called photophosphorylation. The Calvin cycle, these are the enzymes of the Calvin cycle are located in the stroma of the chloroplast, and these enzymes in the Calvin cycle are synthesizing sugars from carbon dioxide, and they use ATP and NADPH that are produced during the light reactions. And this is a cycle that begins with what's called carbon fixation, incorporating carbon dioxide into an organic molecule that is then used to, in a cyclic pathway, to produce larger organic molecules. And we're going to look at the Calvin cycle in more detail on Friday. But I just want to point out a couple things. So here's the structure of NADP+, and it looks essentially identical to NAD, except it's got this additional phosphate group that's on the 2' position of this ribose ring here. So again, this is a dinucleotide, and it's linked together by 2 phosphates, so there's a phosphodiester bond right there. And just as a reminder, this is a linkage that does not occur naturally in DNA or RNA, right? It's a 5' to 5' linkage right here. Instead of if this were in DNA or RNA, it would be a 3' to 5' linkage. This is a 5' to 5' linkage. And there's our friend adenine, so a very common component of these kinds of cofactors or coenzymes. So lots of proteins. You can imagine that when you see that, you know that there's probably some conserved motifs in the enzymes that use this because they've evolved to recognize this same structure, this adenine ring and the attached ribose. So So let's just go through the kind of the overview of what's going on and where this is happening. So this is a cartoon showing the double membrane of the chloroplast, and inside are these thylakoid stacks that are interconnected. So these are all membranes here, right? And the stroma is outside. And so when water enters the chloroplast, in the presence of light, it's inside these thylakoids that the light reactions are occurring. We also need input of NADP plus and ADP plus inorganic phosphate. So these are all the inputs to the light reactions right here. And the output is the production of ATP molecule and NADPH, the reduced form of this NADP plus coenzyme, as well as oxygen. So this is where water is actually getting split, and we have oxygen being produced. Now, if this were going on in isolation, right, it would quickly come to a stop because we would run out of a source of these molecules right here. But instead, this is actually hooked up to the Calvin cycle. So this is a cyclic pathway involving a series of enzymes over here, and these enzymes are located in the stroma, not inside the thylakoid membrane. They're in the stroma. And in this pathway, we have carbon dioxide entering, carbon fixation going on in the first step of the Calvin cycle, and then in a pathway that involves the consumption of NADPH and ATP, we get the production of larger carbon skeletons in this pathway. Right, so this is where the actual synthesis is going on over here. And you can see that these are nicely coupled because the input the output of one set of reactions, namely the light reactions over here, ATP and NADPH, are the inputs to the Calvin cycle. So again, just remember that these are molecules that are storing chemical energy that can then be used over here in this biosynthetic pathway in reactions that require chemical energy. So a lot of those reactions are endergonic, they're energy-requiring reactions, and they're driven by the inputs coming from these molecules that are coming as the products of the light reactions. Then, of course, after they are used in the Calvin cycle, the outputs here are ADP and inorganic phosphate after ATP has been hydrolyzed, and the oxidation of NADPH to NADP+. And so the outputs of the Calvin cycle here are then the inputs for the light reactions over here. So it's a very nice pathway to processes that are coupled through these molecules that are being repeatedly oxidized and reduced in the process. So the light reactions convert solar energy to the chemical energy of ATP and NADPH. And so really we can think about chloroplasts as being solar-powered chemical factories, right? Their thylakoid membranes are transforming light energy into the chemical energy of ATP and NADPH. So they're harvesting some of that energy in a form that can then be used for synthesizing more complex molecules. We can't have one without the other inside of these cells. So if we think about this a little bit more, we have to think about how light is utilized, and the fact that light is a form of electromagnetic energy. So it's composed of, certainly light coming from the sun is many, many wavelengths of radiation. Some are visible, some are not. And these cells and the reactions that are going on inside of them are highly evolved to recognize and interact with just particular wavelengths of light that are coming in. So you know that light is a form of electromagnetic energy or radiation, and it travels in waves. You know that the wavelength is the distance between the crests of the waves, and the wavelength determines the type of energy, right? The length between the waves determines whether it's very high energy, low energy, whether we can see the light or not. And we can talk about the electromagnetic spectrum as the entire range of electromagnetic energy or radiation. And visible light is just a very small section out of that whole electromagnetic spectrum. It's just the region that our eyes can actually detect. And it includes wavelengths that range from about 380 to about 750 nanometers. These include the wavelengths that actually drive photosynthesis, as we'll see. And importantly, light behaves not only as a wave, but it also behaves as a particle, right? Photons. And so this is just a picture that shows the entire electromagnetic spectrum up here, broken down according to the different types that we can categorize according to their wavelengths. And you can see that the visible light is just a very small segment out of this entire spectrum that ranges from 380 to 750 nanometers. And importantly, the shorter wavelengths over here correspond to higher energy radiation, right? Shorter wavelengths, higher energy, and then of course longer wavelengths over here, lower energy. And so when people have been trying to understand photosynthesis and what kinds of radiation are plants able to utilize, then there have been a number of experiments that have been done to try to figure out which part of this spectrum is actually useful to plants. And is it just the visible spectrum, or is it other wavelengths? How do plants deal with radiation that comes in, incident radiation that could potentially be damaging to cells in plants or to molecules inside of those cells? How do they deal with that radiation? And one of the important ways that they do that is that they include a whole series of pigments that are able to, these are small organic molecules that are able to interact with these different wavelengths of radiation in different ways. Some of them actually harvest that radiation, they interact with it productively and can be used in light reactions, and some of them, sort of in response to the question that came up over here, their job is actually to absorb radiation that could otherwise be damaging to plants and prevent it from causing reactions that one wouldn't want to occur that might be destructive to those cells. So let's talk a little bit about these photosynthetic pigments. So these are really the light receptors. These are the molecules whose job is to interact with different parts of the electromagnetic spectrum and either harvest that energy or, as we said, prevent it from doing damage in the cells. These pigments are, does anybody know, what are they? Are they proteins? They're not, right? They're not proteins. They're typically, if you peeked ahead or looked in the book, they're actually organic molecules. Some of them are kind of large organic molecules, but not as big as proteins or nucleic acids or large polymers